resume it and share the screen. Okay, you should be seeing my screen now. Let's go ahead and close that. So I think we'll start here. Today we have a lot to discuss. We have the Lab 01 we're going to do, and I'll discuss that before you get started. But we also need to discuss the infectious disease project. And I told you last time you should look at that file and read it because I was going to discuss it today. Any questions about what we're doing? Okay. So if you come to the Canvas website and then move down to Week 1 Lab, you will see the Infectious Disease Research Project. So this is where it is. It's not due till July 17th, but you're going to need time to work on this. This is a 100-point project. If you click on that link, we'll go ahead and do that. Excuse me. I had a sneeze there. You'll get this, which doesn't really tell you much, but you click that link and it'll get you the Infectious Disease Project instructions. This page does tell you when it's available and when it has to be turned in. And on this page, I think up at the right here, but maybe a button down below, you'll have a submit button for submitting your document. I don't have that because I'm an instructor. I have the speed grader button. Any question about any of that? All right.
So are you assigning us our
topic, this is
or are we picking
I'm going to talk about it.
Oh.
Okay. All right. But yeah, you should take a look at that and read it. I'm only going to go over it briefly because I'm assuming you've read it. And if you haven't read it, you need to read it. So the introduction of the Infectious Disease Project, the rationale of the project, you guys can read about that. I'll talk a little bit about the disease project format, and that is you pick an infectious disease and then check with me, your instructor, about your disease. If no one has your disease, and I agree, you may pick that topic and you may work on that disease. It has to be an infectious disease and one caused by a microbe or a worm. So if you say something like cancer, it's generally no, because most cancers are not infectious and they're not caused by a microbe. Okay, so very rarely do I tell students no, but occasionally students do choose a disease which is not an infectious disease. Then we'll do research on the internet about that disease. You can perform a search of the literature and then start writing up your report. If you get your information from the internet, you should verify your facts on the internet, because anyone can put up anything on the internet. Generally, if you use a college website or a government website, the information will be good. But if you use a student page on a college website, the information could be mistaken. So you need to check your source. And generally for the internet, what you should do is always have two sources of information. If you get information out of a scientific book, like your textbook, or a science book, I don't know, something talking about infectious diseases, it'll be good because that has been reviewed. Magazines, scientific articles, journals, those are reviewed. On the magazine, it depends on the magazine. You'll have to look. If you get something generally like the New York Times, that's not a magazine, but it's a newspaper, it's generally good. But then if you get something like the National Enquirer, it's generally bad, poor quality. So learn to check the quality. If you put up something that's wrong, you will be marked down. You do need to provide references. I'll scroll down to the bottom here. So I'll look at your citations. I'm talking about full references. There must be at least 4 reputable sources. Wikipedia is not considered reputable because anyone can go up there and change the information on Wikipedia. And I and another instructor once did that. He went to Charles Darwin because he reasoned that that one would be watched and that they might prevent him from doing that. And he just went in there and changed the page. Of course, the information he put was correct, but my point is that Wikipedia can be changed by anyone at any time, and so we don't allow that as a reference. That doesn't mean you can't use Wikipedia. It just means if any information you get from Wikipedia, you'll have to back it up by another source. Liza, I'm going to mute you because I'm getting noise here. So let me mention again that I've got everybody muted by default. So if you want to talk, you have to unmute yourself. But then when you're done talking, mute yourself so that we don't get background noise. The only reason why I noticed it was because I heard background noise. All right. The point is that the first person who chooses a topic, you get the topic. The second person who chooses that topic, I am not going to generally allow. I've decided I will allow like 2 people to do COVID-19 if they choose different aspects of the disease to report on. And I will allow that because we're a very large class and there's obviously more than 40 infectious diseases, but there's at least 40 students, maybe more. And I've decided that I will allow 2 people to work on the same disease as long as they choose different aspects of that disease. Any question about that? Okay? So, in a sense, I'm going to be asking you for a disease and then what aspect you're going to be focusing in on. And then do your research, find out about the disease, keep your reference material, because you'll need to write about it, and then write up your report. It's going to be a written report detailing the information about your disease. The report must be typed, single-spaced, 1-inch margin, and it must have 12-point font. My eyesight has gotten somewhat poor, that's why I'm wearing reading glasses, and if it's small print, I'll have a hard time reading it. You should have cover information, but do not have a cover page. Put your cover information on the top of your first page. Does everyone understand that? I don't want your first page to be a cover page because that just wastes space, and if I print it out, it'll waste paper. Put your cover information, meaning oops, there's a mistake. Biology 205. Microbiology, your name, what the topic is. That's all you really need for the cover information. Of course, that won't save it online, but I'll change that. Let me make a note of that. I'm surprised the student didn't catch that. Another change I have to make. I've actually made all my changes, but there's another one I found. And then you'll have the text, the main body of the information. This should not exceed 5 pages, single-sided pages, and then describe all aspects of your disease. This will be worth 60 points, and I'll give you a breakdown of the points. 15 points are for grammar, spelling, flow, readability. The other points will be the specific points, which I'll show you exactly what in a few minutes. You should write as much of it as possible in your own words. So change what you get out of Wikipedia and put it in your own words. I will be doing a plagiarism search of this article, and anything that is over 20% in common with something else, I'll look at further. Anything you use from another source directly, you got to put it in quotes and say where you got it from. And then furthermore, if you do that, that should be one of your references. Okay, so you should have an in-text citation for that information, and then have a full text citation at the end. Plagiarism will result in a score of 0 on the paper. You will have to have in-text citations in this paper, and in-text citations are worth 10 points. You may use any commonly used citation format. I think MLA was the one that you have for the plagiarism quiz, but you can use any other format. But make sure you're using all of that format throughout the paper. If you don't know how to do an in-text citation, generally you give, if the article is written by one person, you give the article's last name and then a comma and then a year. So you would go parentheses, author's last name, comma, year, and then parentheses. Okay? That's generally the way you use an in-text citation. And then that author will be in your full references at the end of the page, and that's where you give the complete references, meaning the author's name, the year, the full name, by the way, the article's name, and then the article's source. If it's a scientific journal, you give the journal name, the volume number, and the page number. If it is from the internet, you give the web address that you got the site from. And that's the full citation, not the in-text citation. All right, so that's the full citation. How you give a full citation will depend on the format where you're getting the information. Like I said, these should have 4 good sources, like a government page, a scientific journal, a popular but good scientific magazine, science books and textbooks, newspapers from a credible source, and then a variety of scientific reputable like television shows or websites. Sources to be suspicious of include religious sources, politically sources, privately funded publications and websites, people's personal websites, college and university pages that contain student work, and like I said, Wikipedia is a susceptible source. The literature citation page, I've already talked about that, but it's going to be worth 15 points. So in your body of your text, you should have subheadings, and one of them will be the causative agent, where you talk about the causative agent. And if it's a bacterium, you say, what are the characteristics of the cell? What's its gram stain? What's its cell shape? What's its growth arrangement? What are its metabolic capabilities? And other things known about that bacteria. Now, obviously, that would be only if it's a bacteria. If it's a virus, you do something similar, but for a virus. If it's a prion, you tell us about the prion. If it's a eukaryote parasitic infection, then you tell us about the parasite. You then have another subsection telling us about the history of the disease. Describe changes in our knowledge and attitudes towards the disease throughout history. Now for COVID-19, the disease has no history, and so that would be something you'd state. You tell us about the epidemiology, describe the prevalence and transmission of the disease. Where in the world is the disease prevalent? Like malaria is found in the tropics. How many people are currently infected? What's the rate of new infections, etc. And then have another subsection talking about the pathology of the disease. And then you have another subsection talking about the response and treatment of the disease. And then lastly, you have the socio and political economic considerations. Describe any historic and present day social and economic or political issues that either help or hinder us in limiting the spread of the disease. What is the cost of the treatment? Like for example, COVID-19, the cost of getting the vaccine is nothing. The insurance company will take up the cost, and then they will bill the government. So the government is paying for the cost of the vaccine. Now obviously if you're talking about the medications, the medication treatment for COVID-19 will have a cost associated with it. Suggest policies and practices that can be employed to help with limiting the spread of the disease. And here is a rubric that I'm going to use for grading the paper. 1 point for the cover information, 9 points discussing the causative agent, 10 points for discussing the history, 10 points for the epidemiology, 10 points for the pathology, 10 points for the response and treatment, 10 points for the social, political, and economic issues, 15 points on the quality of the writing, 10 points for your Intext Citations, and 15 points for your literature citation page. Let me warn you that students often get a subtraction for not doing the Intext citation or the literature citation page correctly. And the total project will be worth 100 points. Any question about it?
I have a couple of questions.
Okay.
My first one is, with the in-text citations, do you prefer direct quotes or can we paraphrase and cite it?
Okay. You can paraphrase, but remember, if ever you use more than 3 words from the original source, I'm talking about 3 words in sequence, obviously you can break them up, and that doesn't count. But 3 words in sequence, it has to be in quotes. So if you paraphrase, make sure you paraphrase the entire sentence. But yes, I prefer you to actually write it in your own words. Okay?
Okay. Okay.
All right.
And also, how do we reach out to you regarding picking our topic? Do you want us to just email you?
Yes, use the Canvas website. I will try and check that Monday through Thursday every day. I'll try and check that. My other emails, I will not check them every day because I get a lot of emails in even the Yavapai email site, which are of no interest to me and they have no bearing on me, so I'm not going to check that site every day. I'll check the Yavapai, what is that? J Volpe at instructor at yc.edu. I'll check that frequently, like several times a week, but I'm not going to check it every day. The Canvas webpage, I will try and check that every day, Monday through Thursday. I'm not going to guarantee I'm going to check it every day, Friday through Sunday, but I will guarantee I'll check it at least once during that 3-day time span. Okay?
Sounds good. Thank you.
Okay? And like I said, just tell me what your disease in and then what aspect you're going to be focusing in on. Like somebody last term wanted to do the flu and they wanted to talk about the H1N1 Spanish influenza that hit the world in the winter of 1917-1918. That's highly specific. You just need to do something like the flu, and you want to do it on, I don't know, H1N1 as opposed to, what's another one, H1N7, something like that. Okay? And don't ask me what H1N7 is. I just pulled that out of my head. I know H1N1 is sometimes called the Spanish influenza and is also sometimes called the swine flu, and it has some other names. All right, so that's the Infectious Disease Project. Any other questions about that? All right, I'm going to close that down, and now we're going to talk about Lab 1. So there's some pre-reading you should do before you do Lab Module 1, the ubiquity of microorganisms. The primary learning objectives upon successful completion of the lab, you should be able to know that microorganisms are almost everywhere. You should be able to describe the purpose of culture media. You should be able to describe colony abundance, diversity, and morphology. And you should be able to define the following terms: inoculation, ubiquity of microorganisms, sterile, isolation, incubation, in vitro, culture media, such as a broth, an agar plate, an agar slant, and an agar deep. You should know what a fastidious organism is. You should know what a non-selective media is. We'll talk about that today. And what ubiquitous means, as well as what a colony is. Any questions about any of that? All right. You should know that microorganisms are organisms that are too small to be seen with the naked eye. And these include bacteria, virus, and prions. And we are also going to include helminths, worms, in our studies, even though the adults are not microscopic. The phrase ubiquity of microorganism refers to the concept that microorganisms are essentially everywhere. In the lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We will also learn basic lab protocols, including the use of solid media, liquid media, although we won't do that one today, labeling lab cultures, procedures for disinfecting lab benches, and procedures for washing our hands. A culture medium is also called a growth medium. It is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms. Culture media may be liquid, and these are called broths, and are commonly used in test tubes. We also add agar to the liquid medium to result in a solid medium. And we can add other things, but agar is the number one thing we add to convert a liquid medium into a solid medium. All right, so there's a video you'll need to watch about preparing pour plates. We're going to be talking about general purpose media. These are non-selective media, and they are usually complex in composition. They support the growth of most non-fastidious organisms using peptone and yeast and beef extracts. So they grow a large number of organisms, they're cheap, and they're easy to use. We tend to use them in microbiology largely because they grow a large number of things and they're cheap. There are 3 general purpose media that we will be using throughout the semester. There's nutrient agar with yeast extract, and that we abbreviate NAYE, N-A-Y-E. There's tryptic soy agar, and that's abbreviated TSA. And then there's nutrient agar, or NA, and it's the same as NAY, it just doesn't have the yeast extracts added to it. All right, any question about general purpose media? Everything you use in the lab has to be labeled, and that includes Petri plates and test tubes. On the labeling, you should add your name, the date, the type of medium it is, such as NA, and then the incubation temperature is optional. You should also put what you have growing, so the inoculum. If you know what it is, put what it is. Like this one, we're growing E. coli, and so we label it E. coli. If it's something that you don't know exactly what it is, put where you got the inoculum. Like today, you'll be virtually, I mean, you're not actually going to do it, you're going to pretend you're doing it virtually. You're going to be exposing plates to the air. So what you would write would be air, or air exposure, because that's your inoculum. Use a piece of tape generally to write on a test tube, and then write on the tape. On the plate, you can write directly on the plate. You should write on the perimeter of the base of the plate, meaning right on the bottom of the plate. The reason why we write on the bottom of the plate is because if the plate gets dropped or some other accident happens and the lid gets removed from the plate, the agar and the bacteria are on the bottom, and so you should always write on the bottom of the plate. Any questions about that? I once worked in a lab, and the technician had a whole stack of plates, let's say over 300 petri dishes, and she had written everything on the lids. And then she was carrying all these plates on a tray, and she tripped, spilling everything onto the floor. Well, the lids got separated from the bottom. And then nobody knew what the bottom of the plates were. And so the whole experiment had to be thrown away and then repeated because she simply did not write her information on the bottom of the plate. Any questions about that? All right. An inoculation is the placement or transfer of a microbial specimen into the medium. Incubation is the placement of the inoculated culture media into the appropriate physical environment for growing the microbes. Generally, we incubate things in an incubator. Occasionally, we'll grow them at room temperature, but generally, most things are incubated in an incubator. Let me show you a picture of an incubator. Here's an incubator. Incubators control the temperature and add humidity to keep the plate from drying out as quickly, meaning it still will dry out, but it will keep it from drying as quickly. Our incubators in the microbiology lab are pretty simple, but you can change the oxygen level as well as the CO2 level in a more complex incubator. You can also change the humidity and specify directly what it is. Ours are much more simpler than that. We just make it more humid inside the incubator than outside. And how we do that is we add water to the incubator. You don't need to know actually any of that. You just need to know a little bit about what an incubator is. During incubation, an agar plate will have water come out of the agar and then condense on the lid of the plate. Therefore, when we incubate plates, we incubate them upside down with the lid on the bottom. I don't know if you can actually, it's having a hard time seeing that. Yellow for some reason doesn't show up well. Let me try this. Of course, there's no real top. Boy, that doesn't show up at all. I never knew that different colors show up differently. Let me try this. It's red. Let's see if red shows up. Yep, red shows up. So this is the top, and this is the bottom. You can't see the black. That's funny. You can't see the bottom, which is, I thought, red. It's not showing up well. For some reason, the top's showing up better than the bottom. Anyways, what we do is we incubate this upside down. There it's partly showing up. And that way, when the moisture comes out of the agar, it will condense on the top, which is where the agar is, and then the agar will whip the moisture throughout it, and so a drop will not form. And even if a drop were to occur and drop on the lid, we say, so what? You have no bacteria on the lid. If you were to put the plates in the incubator right side up, with the lid on top. The moisture would condense on the lid and then drop down in the bacteria. What's the word I'm looking for? Dispersing the bacteria. And so we want to prevent that from happening. So we always incubate plates in the incubator upside down. When we have growth on the plate, the bacteria colonies tend to have a rather smooth, textured surface. Although there are exceptions, the majority of them, and this is over 90% of the bacteria, have smooth, evenly defined colony borders. In comparison, fungal colonies tend to be furry or hairy. So this is one way we can tell bacteria colonies from fungal colonies. You should know that each colony is made of tens to hundreds of millions of cells, hopefully all descended from one cell that started the colony. So here we're looking at colonies. Let me see if I can blow this up a little bit. That colony looks fuzzy. The border is not sharp. If you look at this colony, and that colony, and that colony, the border is very sharp. There's no question about where the colony is and where the bacteria is. And I don't know how to mark that. So I was saying I bet there's a way for me to mark that, but I'll just do it with the mouse here. And this colony, it's not sharp. It's fuzzy or hairy. This one, the black, is very fuzzy and hairy. The white, it's not showing, but it's also fuzzy and hairy. These are fungal colonies. They tend to be bigger also than bacteria colonies. And that's how you can tell a bacterial colony from a fungal colony. That works for over 90% of the species. All right, any question about any of that? There's a video on watching bacterial cells grow by binary fission. Please watch that when you're going through the lab. And you need to know about colony characteristics. You'll be pleased to know if I ask you a question on a quiz or exam about colony characteristics, I will provide this chart. But you do need to know how to use the chart. You can talk about the shape of the colony, whether it's circular, rhizoid, irregular, etc. You can talk about the margin. By margin, we mean the edge of the colony. So this is an entire margin. This is an undulate, a lobate, a rhizoid. And if I'm going to ask you about how to delineate these, I will not ask you to separate undulate from lobate and curl, because that's too difficult. I would ask you to separate entire from one of these three. And I would ask you to separate these two from these three and from this one. Okay? Any question about colony characteristics? Colonies have characteristics like a specific shape, a specific margin, an elevation. That's how high the colony is. I probably will not ask you about elevation, because to see the elevation, you really have to take the lid off the plate and then look at it from the side. But you can have a flat colony, a raised colony, and that's a mesa, a convex colony, and an umbonate. I will not ask you to separate a convex from a pulvinate. That would be too hard even for me to do. A pulvinate is just more extreme than convex. The size, there's punctiform. And let me give you an example of punctiform. Punctiform, that pencil's not showing. Ah, there it is. If I put it by my face, it shows. Punctiform means it's the size of the point of a pencil or smaller. Small is about the size of the point of a pencil, and I'm talking about a somewhat dull pencil. Moderate is between the size of the point of the pencil and the eraser of the pencil. If it's large, it's the size of an eraser of a pencil or larger. And then there's the texture of the colony, the appearance, the pigmentation. By pigmentation, we break this into colored, like purple, red, yellow, and that's pigmented, and then non-pigmented, which is uncolored. And that would be a cream, a tan, a white, an off-white colony. And then the optical characteristics of the colony. Most colonies are opaque. Some are translucent. Very few are transparent. So if we look at these colonies here, that colony is opaque. That colony is opaque. This colony, at least the edge, is translucent. That colony is probably translucent, and that colony is opaque. I don't see any colonies here which are transparent, and a transparent colony is difficult to see because you look through it like it was water, meaning you can see right through the colony. Few colonies are transparent. Anyways, this is giving you the different characteristics of a colony. Colony A is large, pale yellow in color, circular shape, basically circular. That's a little off circular right there, but that could be because of that colony. Shiny, pulvinate, that I can't see at all, and the margin is entire. When we're talking about colonies, we can talk about their abundance and their diversity. Abundance is simply the total number of colonies on the agar plate. So you simply count them. Excuse me here. It's starting to hurt my ear. Diversity is the number of different colonies on the plate. We can't be entirely certain of the diversity because when we look at colonies, it looks like that colony, it looks like that colony is the same colony as that colony. Can you guys see my mouse? Can somebody see the mouse? Can you shake your head, Ariana?
Oh yeah, I can see it.
Okay, good. So it looks like these colonies are the same, but that colony looks different because it's darker than these colonies. Okay, so it's sometimes difficult to determine the exact number of diversity, but you go on the colony characteristics and you generalize. Like that colony looks different from that colony. Not only is the size very different, but the coloration is slightly different. The opacity might be slightly different too. That colony looks different from this colony because the coloration is slightly different, and then the margin is very different on this colony compared to that colony. Any question about that?
I do have a question.
Huh?
So with that figure 5, the colony characteristics, for example, if you had a bacterial colony, so are you saying that could take on any of those shapes, margins, elevations?
Yeah, the different colonies can take all of those colony characteristics. Did I answer that?
But it's not like specific to fungus, or say like bacteria are not always going to be circular. They could be any of those other shapes.
Correct. That is not circular. Neither is that, or that. Bacteria can be any of these shapes. Okay? The most common are circular or irregular. The others are uncommon. That is irregular. This colony. Let me blow it up. Let me see if I can find another irregular. Oh, that didn't work. Well, there's an irregular colony. That one is slightly irregular, but most of them are circular. Like that one's circular, that one's circular, that one's circular, that one's circular. At least in this picture.
So is there something that's determining what's changing those shapes?
Yeah, the different species will have different shapes, different colony characteristics. And that's why we can use some of those characteristics to figure out what are different species and determine the diversity. The diversity is the differences or different types of colonies. Like in this picture, what's the abundance of wild cats? Come on, people. The abundance of wild cats. I got the definition right there.
7.
Yeah, there's 7 cats. 1, 2, 3, 4, 5, 6, 7. And what's the diversity of cat species?
2.
2, lions and cheetahs. Thank you. All right, that's it for the lab manual. So I'm going to tell you that I don't have that there, so let's go to the web page. Click on this link to get lab manual or lab worksheet 1 and fill it out. Click on that link to get it and then submit it. You'll have a button here at the bottom of the page for submitting it, and then submit that by 11:59 p.m. this Saturday, June 12th. And that's it for Lab 1. Any questions? All right. If there's no questions, work on the lab. I will be here to answer questions until... where are we? I think not very long today because the talking took so long. Until 3:45. All right. So get started on the lab.
I do have another question just looking through
Okay.
this. So for example, procedure 5, are there microorganisms in the soil? And so it's as if we've already inoculated, like incubated this, and that's what we see then?
I'm not sure I understood the question. Can you repeat that?
Well, it says in procedure 5 to collect some soil, which is obviously we're not doing that.
Well, yeah,
And so-
everything you're doing will be done online.
We're just looking at the picture, and that's basically the result that we got. Is that correct?
Correct. Okay. Maybe I didn't understand the question, but everything you're doing is online. So we're providing the results to you. You're not actually going to be doing any of the lab in a lab.
Okay,
Okay?
I might sound like kind of stupid, but how do I figure out what's in the air? Like, where is this information at?
It should be in the lab. So let's go to the question. Which question are you referring to?
Question, I'm so sorry. This is all new to me. So like, okay, what is the procedure
All right.
one?
What is in the air you breathe? You don't need to answer. You do need to answer, though, procedure 1, A, B, and C. Okay? So what colony abundance is on this plate? Count the colonies. What's the colony diversity on the plate? Determine what are different colonies, and then count the different number of colonies. Okay? What step would you take to reduce airborne contamination of a sterile agar plate that you are inoculating? This one you have to think about, but it is mentioned in the lab. So take a look. If you don't know the answer, take a look in the lab module for today. I still have that up? Yeah, it is here. Okay? And then write in your answers, and then submit your answers. Now, if you're worried, that's why I'm here until 3:45. Another thing is I consider the lab to be, I don't know, a training exercise. And so when I grade it, I will tell you procedure 1, step B is not correct. And then you will have a few days to submit a correction. So in the lab, you will have 2 attempts to get the right answer. I'll word it that way. And that's because I consider the lab to be a training exercise. So the lab worksheets, you will have 2 attempts to get the correct answer. And I'll tell you which ones are wrong. I won't tell you what the answer is, but I will tell you which ones are wrong. In the lecture worksheets, if you don't know the answer, because generally you're only going to be answering 1 or 2 questions, you should look it up, because that will be graded. And you only have 1 attempt to get the right answer. But on the lab worksheets, you'll have 2 attempts to get the correct answer. Of course, you have to look at it and then make the correction by the due date. Generally, the labs are due at 11:59 p.m. Saturday night. If I get them graded on Sunday, I will generally say that the correction has to be made by Tuesday, because I don't like the labs to overlap. And most people won't start the next lab until Tuesday. But if I don't get it graded, let's say until Tuesday morning, I will allow you to make the correction by Saturday. Okay? So it depends when I get the grading done, when it'll be due. But sometimes it'll be due on Tuesday, and sometimes it'll be due on Saturday. And if I get it graded on Monday, I might have it someday between those 2 days, like Thursday. Okay? Any question about any of that?
Yes, I have a question. So you're mentioning the lecture worksheet. That's just one question that we're answering, and then everyone else is also providing an answer, right? We're not turning in the entire worksheet completed.
On the worksheet for the lecture, you're generally answering only 1 or 2 questions. We're a very large class, so I suspect it'll generally be 1 question. And really, we're 2 classes into 1. Okay? Any questions about that? All right. If there's no further questions, go ahead and get started on the lab. I'm going to stop the share and stop the video